Hello and good afternoon, everyone. Today we are going to experiment on mulberry plants. So the first thing that you have to do is cut the plants. You have to cut the plants by counting 4 nodes and cutting below the 4 nodes. Just like that, and you have to cut it in slanted form. Cutting in slanted form is to increase the surface area for absorption of nutrient. Next, you have to separate the stems and cut the stem. Make sure it is pencil size. You have to count 4 nodes, 1, 2, 3, 4, and then cut it above the 4 nodes that you counted, slanted. Just like that. And then that's your pencil size stem. Next, you can repeat all the processes by counting 1, 2, 3, 4 knots and cutting it above the knots that you counted slantedly. Just like that. And now you're done. But before that, you have to remove all the leaves using the tool that you have, or you can also hand pluck it using your hand. And here are the processes that we repeated to get all the stems that we need for the experiment. Here you can see these 2 soils in separate container. One is for the stem with the auxin and one without the auxin. To prepare the soil, you need to water the soil and make sure that the water is evenly distributed throughout the soil. This is to ensure that nutrients are absorbed into the soil and the stem will get enough nutrient for root growth. Not only that, you can also use this tool to make sure that no dry soil is incorporated into the soil. If there is not enough water, you can add more water to the soil and mixing it all together. By doing so, we can ensure that the moisture of the soil will be retained and root growth will not be stunted. Next, you will have to repeat the process with the soil in the container without the auxin. So here, now you have to prepare soil ready for the stems to be planted. You take your pencil-sized stem. Here is your pencil-sized stem. Here is the top part and here is the bottom part. You have to dip it in at the bottom part for root growth and not the top part. First, you take the water. You dip the stem into the water to make sure it is wet and to ensure that the auxin will stick on the stem. Here's the auxin from the lab. To dip it, you have to open the lid first. Be careful when opening the lid. Once it is open, just dip the stem into the auxin and tap out excess auxin to prevent wastage from happening. Now, just dip the stem with the auxin and make sure that the knot is above the soil. Just like that. And the one without the auxin, you can just dip it into the soil and make sure that the knot is above the soil. The steps are repeated with the remaining stems. With the auxin, we just dip it in the water and auxin, and without the auxin, you just dip it directly into the soil. Make sure the knots is above the soil when dipping it into the soil. Here are the processes without the auxin. As you can see, we are just dipping it directly into the soil, dipping the stems directly into the soil. And here are the processes of dipping the stems with auxin into the soil. And here are the finished products. So here are the stems after 3 weeks of growing. As you can see, they have different roots. The one with the auxins have longer roots and straight roots, which follows the gravity towards down due to the available of auxin. And without the auxin, the roots are curling up and not following the gravity compared to the one with the auxin. Next, we will be reviewing the color of leaf between those 2 stems. Now, with the auxin, the color of leaf appear greener and also it has more leaf to be compared to the one without the auxin. This is because auxin promotes elongation of cell and more leaf are produced.